Joanna. Okay, thanks.
So Sunit, you want to get us started? So just a little context here. I know we talked a couple meetings ago about reminding ourselves of why we're doing this and the importance of funding research. And just to remind us, we have these great researchers, we've been able to fund grants, and so we fundraise to be able to continue to provide grants, to continue to be able to do this research. So we want to let you know what your fundraising dollars have gone to and what the grants have provided and supported. So we've asked them to come and tell us what they're doing with their grants. So Sunit, I'm going to ask you to start us off.
Great, thanks so much. Great to see everyone. So I'm always excited to tell you what your research dollars have funded, because in both instances, they've been incredibly impactful, and the impact continues on even after the award is over. So the first one, I almost forget to talk about this, but it's like the first one that was given that was so important, that was basically to expand a single center bone marrow transplant trial to a multi-center trial. That's for a reduced intensity conditioning regimen for dyskeratosis congenita that started at our center in 2012 and then was expanding in 2016, and then under the funding essentially that enabled that from Team Telomere and the MDBR. And so I think just to fast forward, that added a few sites and really provided an impetus for expansion, which is definitely a barrier in clinical trials. And now the protocol continues to be active. We've treated 30 patients amongst the consortium at this stage, and many other patients off protocol at centers that just couldn't join for a variety of reasons. And we have 15 centers. It's international. We have about 6 or 7 centers, actually almost too many centers now, pending for our current bandwidth. And we have a current goal of trying to accrue 40 patients for the second phase of the protocol. And we probably will just continue to expand it after that. We're actively thinking about that right now. The group in Brazil wants to join, and that's 5 centers in Brazil. So when there's only 10 spots left, you have to really think carefully about how you're going to do that. So it's really, really good. And we have an intent to make sure that that's formally published beyond conference abstracts at the present time. It's definitely been disseminated in international conferences, and really try to apply for a real definitive funding to study the biology alongside the protocol too. So that's the first award that you provided. And the second one, again, the one that's more actively on my mind these days, as I think beyond bone marrow transplant, was how do you come up with a systemic therapy for dyskeratosis congenita? And so the MDBR funding provided leverage to take something that was an observation in the lab and an idea about a small molecule approach to treating, small molecule being like a drug, hopefully for treating dyskeratosis congenita. And essentially the scope of that award was to try it out on a variety of patient genotypes and see how broad the effect could be of that too. And so we have a little bit of a good news, bad news thing in that sense, is that we definitely got to explore a scope of genotypes, and it definitely seems to work in the ones that we were targeting initially, like PARN and DKC1 and some TERT mutations. And then it showed us clearly that it may not be really something that we can demonstrate to be effective in other genotypes. And so that tells us, yeah, keep on focusing on trying to find other strategies for some of those other genotypes as well. But that has been quite successful so far, and it was published last year, and then is garnering what we essentially need in this phase is for that to actually attract additional investment from either non-traditional funding sources or from partnership. And the latter hasn't quite happened yet, but we did get a translational grant that was based on that research from the NIH for $1.2 million. So we're using that to actually keep that going and try to get a partner. We really need a partner, so we're going to try to get a partner, and that's I'll update you when that happens. So I'm excited to do that soon. Anyway, so that's the update. Thank you very, very much for the research support. It's been invaluable, and hopefully will turn into therapies beyond just the blood.
Wonderful. Thank you, Sunit. Thanks for working for us. Brad, you want to jump in?
Yeah, I actually have a few slides, so I'll share my screen. But let me just say first, I'll talk about 2 funded groups of studies. One is headed by Dasha Babashuk, who's an assistant professor here at Penn. She couldn't be here tonight, so I'm doing this for her. I'm peripherally involved, but it's mostly her work. And that was funded, not this most recent round, but the year before. And then I'll talk briefly, again with some slides, maybe more than you want to see, but about longstanding projects that I've been undertaking with Chris Lengner's lab and some other labs that we also got support from Team Telomere from years ago, and which has turned into NIH-funded projects at this point. Let me start with Dasha's work. So share screen. Do you see the PowerPoint?
Yes.
Okay, so Dasha sent me these slides about an hour ago. I added her picture. She wouldn't be vain enough to add her own picture to her presentation. I know her. But the title's Pre-Malignant Clonal Evolution in Telomere Biology Disorders. So the idea here is that, as you know, people with these disorders are more likely to get certain kinds of hematologic malignancies, for example, AML. And we're trying to understand, what the basis for that might be. And one concept here is that you can see in the context of these diseases that there's so-called clonal hematopoiesis. So what that means is that basically one cell that's experienced certain kinds of problematic mutations is now expanding and taking over the population of cells in the blood. And that's basically one key aspect of cancer, these clonal expansions. Those have been studied in the context of malignancies in people who don't have telomere biology diseases. And they're driven by mutations in particular kinds of genes. Actually, Tim Olson, who's also participating in this study, has studied that in the past in non-telomere biology disorder, and for example, seen mutations in HLA genes, which are involved in the immune system. Anyway, Dasha has looked a little bit in telomere biology diseases, and she doesn't see the same mutations. And so she's hypothesized that there'll be a different set of mutations driving these clonal expansions in telomere diseases. And in particular, I think very reasonably, she's hypothesizing that there'll be mutations in proteins that normally respond to telomere dysfunction. So the idea is that the reason we even have telomere dysfunction, even in normal people, is as a way to stop cancer. And that works pretty well, not perfectly. People still get cancer. And in people with telomere biology disorders, it's actually maybe even turned toward the negative side of things, that the telomeres become so short and dysfunctional that instead of just stopping cancer, they can actually drive cancer. But nonetheless, when the telomeres become dysfunctional, there are a bunch of proteins that recognize that telomere dysfunction and stop the cells from becoming cancerous. If you get a mutation in one of those sort of guardian proteins or guardian genes, then you get the cancer. And so she's looking for mutations in those kinds of guardian genes. And she and I participated in this, came up with a list of the likely genes where these mutations might reside. And she's basically sequencing in a directed fashion, trying to look at these particular candidate genes to see if you can see mutations. She's also doing some gene expression profiling in blood from these patients to just try to understand the biology of the malignancies that might be unique to these telomere disorder patients. So this is her progress so far. She has a cohort of 44 patients. She's designed this panel of genes to look for mutations in, and actually done all of the sequencing. And at this point, she and her group are going through all of the sequencing data, sort of with a fine-tooth comb to see if mutations are there. And she's in the middle of it, but says things are going well. And we'll do a bunch of tests to confirm that these are really driver mutations. She's also begun the gene expression profiling experiments using a new kind of technique called single-cell RNA sequencing. It's possible to just look one cell at a time at which genes are expressed. And that's useful in this particular context because there aren't a lot of cells that you can get from these patients. It's useful for other reasons too, but that's ongoing. So that's Dasha's project. And then the projects I've been carrying out with Chris Lengner's group. Let's see. I think I need to stop sharing. Actually, are there any questions about Dasha's project? Okay. This is a little bit less clinical, the projects I'm undertaking with Chris Langer's group. Instead of looking at direct patient samples, we're taking so-called adult pluripotent stem cells, iPSCs, that are either normal or have mutations that can cause telomere biology disorders. And we're taking these cells and treating them with different cues in culture that can turn them into different kinds of tissues. We started doing this in the gut and the intestine and learned a lot, but now we've moved into the liver and the lung. And the idea is to understand what the mechanisms might be that drive diseases in these different tissues in people with telomere disorders, and then also to test potential therapeutic approaches. And one of the things we learned in the intestine was that there seems to be a feedback loop between this Wnt pathway, which is a way that cells talk to each other, and the capping of telomeres. And this is a pathway that we could potentially intervene in pharmacologically. And we now have data on the lung. We've submitted this to a journal, eLife, which is a solid journal, and got good reviews, and we're just finishing up some final experiments. So this should be published soon. But this was primarily the work of Rafi Fernandez, an MD PhD student in my lab. And basically what we found is that if we make lung cells, a particular kind of lung cell called an AT2 cell, which is probably the most important cell for pulmonary fibrosis pathogenesis. If we do that with a mutation in DKC1, the telomeres get short, and eventually these cells lose their ability to proliferate, similar to what you see in other cell types. They still differentiate though. They still become lung cells, but they can't divide. They show signs of what we call senescence, which happens in many cell types in response to telomere dysfunction. That's what this is all about. And then interestingly, just like we saw in the gut, we also see signs of abnormal Wnt pathway activity. So it seems to be a parallel kind of biology to what we saw in the gut. There's a lot of information here, but for example, this graph shows the expression of different Wnt proteins. These proteins I said can talk between cells, and some of them go up, some of them go down. We can't say from this whether the Wnt pathway is up or down, but looking sort of broadly at changes in lots of genes in these artificial lung cells, it looks like the pathway is going down. And I think a clearer sign of that is that the receptors for these Wnt proteins, so Wnt's are made by one cell and they go to another cell and they're received by these frizzled receptor proteins. They're solidly down, okay? And then on top of that, if we pharmacologically interfere with Wnt signaling to kind of enhance it, actually interfere is a bad word, but if we, it's a misleading word, if we inhibit an enzyme called GSK3, which is an enzyme that Wnt normally inhibits, we can restore growth in the mutant lung cells and restore telomerase activity in the mutant lung cells. Really interestingly, the mutant cells seem more responsive to an elevation in telomerase, or they respond by elevating the telomerase even more than normal cells in response to the therapy. We don't understand the basis for that at all, but it's kind of exciting. It's like the mutant cells are hungry for a signal to make more telomerase. And somehow that seems to be happening in this system. I want to mention really quickly that we've been collaborating with Andy Vaughn's lab, who's also at Penn. He's a lung expert. And he, for the first time that I'm aware of, has been able to take human lung cells, the ones we make in our lab, and transplant them into mice. And they actually engraft into the lungs of the mice. So it's kind of like you have these little islands of human lung inside the mouse lungs. So it's not a perfect human lung, but we think this is a model that we can explore to understand how the telomere dysfunction in the human cells that we put in might be driving lung disease. And we've gotten to the point where we've now put in either normal lung cells or lung cells with dysfunctional telomeres. But we don't know yet whether those dysfunctional telomere lung cells are creating any kind of disease in the mouse lungs, but we're right in the middle of our first round of looking at that. So I'm very excited about it. I mentioned we're doing it in the liver too, just real quickly. This is mostly being carried out in Chris Langner's lab. Young Jun Choi is a postdoc doing the work. And we found a result that's very different than what we saw in the gut and in the lung, okay? This is, I think, kind of bad news from the standpoint of the idea that we can just augment Wnt signaling and it'll be good across the board if you have a telomere disease. But it also says that it's a good thing we're exploring this carefully in different tissues to try to understand the biology. And in particular, what we see is that in contrast to the lung and the gut, liver cells with telomere dysfunction become hyperproliferative, they divide more. And actually the Wnt pathway is more active in this context. One really cool experiment that Chris's lab did was to mix mutant hepatocytes, sort of the main cell in the liver, with so-called stellate cells. These are the cells that make extracellular matrix that can drive fibrosis, like cirrhosis, the problems you get in the liver with telomere dysfunction. And the result that Young Jun is finding is that it's the genotype of the hepatocyte that matters. If you mix a mutant in red hepatocyte with a mutant stellate cell, you get a large, unusual organoid. If you put in a normal stellate cell, it's still unusual. But regardless of the kind of stellate cell you have, if the hepatocyte is normal, everything's fine. So that it's just teaching us about the biology a little bit. I will skip this because I think I'm going too long. I'll just mention it real quick. One of the things that's for us in the lab difficult is that when we introduce a mutation in a gene that normally supports telomere maintenance, we don't know when the telomere dysfunction is going to happen. It's just like in the patients. Like if you inherit a mutation in a gene that we know causes dyskeratosis congenita or another disease, the age at which you have problems or the particular tissue in which you have problems isn't trivial to predict just based on the mutation. It depends on the telomeres that you inherited and other factors. So we've made a new system with these iPS cells that we can turn into tissues where we can induce telomere dysfunction on command. And Katrina Stepp, who's shown here as a graduate student in my lab, who's built this system. And I'll spare you the details, but basically we're excited to be able to basically add a molecule and get the telomeres to uncap immediately. And we think this will give us more clues to what's going on and more power to understand the mechanisms. So that's it.
You're busy. That's a lot, thank you.
Yeah.
And then Judy, who's our most recent recipient, we'd like to hear about how your research is going.
Okay, I wish that I actually go before Brad because he make a very detailed introduction to what I'm going to talk about. I will also share my screen, but my project is, of course, at its infancy. We actually created some cell models and hopefully we'll be able to start studying them soon. Would I be able to share my screen?
Yep. So Brad, if you want to,
I'm still sharing. Sorry.
Yeah, no, you're not sharing. I just made you
Oh.
the host. So you have to go up
Okay.
to Judy's 3 little dots and make her the host, please, in
Okay.
her right-hand corner.
Let's see. Make host. Okay, Judy, you should have
Thank you.
control.
Let's try this. Share screen. Advance. Okay. Can everyone see my?
Yes.
Okay. So very briefly, I'm going to talk a little bit about telomere dynamics, just to bring everyone into the same page. As everyone knows, telomere biology disease is failure to maintain what is needed throughout our lifetime to keep the telomere length of somatic of our stem cell as well as proliferative cell compartment in a capacity that allowed us to have renewal of these tissue. Telomere length are constantly shortening in somatic human cells, and as they go through this cycle of shortening, they reach a time point where the length becomes too short to have a protective function at the end of the chromosome, leading to the proliferative arrest, meaning that the compartment of cells cannot continue dividing anymore, and therefore you lose tissue integrity, and in the hematopoietic tissue, it leads to bone marrow failure. So in a number of cases, when this proliferative arrest is actually not obeyed, meaning that there are some surveillance mechanism failure, the cells that are having short telomeres can continue to divide. And this continued division would allow these cells to become genomically unstable, meaning that their genes started to cut and paste together, leading to abnormal number of chromosome and number of other genetic alteration. And that is requirement and the necessary event that leads to the cancer initiation. So this cancer initiation process is happen much more frequently, as we can imagine, with telomere biology patient, because their telomere are shortening at a much faster rate. However, in order for a cancer to proliferate indefinitely and leading to the actual malignant stage, they have to restore the capacity to maintain telomere. So that's actually where the mystery comes from. When telomere biology disorder patient actually cannot upkeep their telomere effectively or optimally, what happened in this transition state that allowed their cancer to become malignant and therefore becoming a clinical problem? So Brett is actually going upon this question using patient sample. But in the case of telomere biology patient, we know that many of them succumb to bone marrow failure as well as organ failure. So the number of tumor sample as well as cancer patient with telomere biology background, it's not that high. So I'm actually very impressed that you have 44 in your cohort. So it's actually very good resources. We might actually hit you up on it later on and look at what your profile looked like. So this is, sorry.
That'd be great. Yes, please do.
Yeah. This is pretty, well, it's the old statistics, but actually it's all we have so far from the inherited bone marrow failure clinical cohort that Sharon Savage is heading. So in 2018, they put out an interim report on all of their collection in their patient cohort and how many of them were actually surviving the bone marrow or hematopoietic dysfunction and going on to develop cancer. And the conclusion of their analysis is that telomere biology disease patient had a much higher risk, log unit higher risk of developing cancer, primarily of hematopoietic as well as head and neck cancer, particularly of the tongue. So we want to know what happened to the mystery. Telomere biology disease means that the patient cannot keep their telomere very well. So how did the cancer develop into a more competent state of developing adequate telomere maintenance, allowing them to proliferate indefinitely? So what we did is instead of looking at clinical data, there are a very old method of turning normal human cells into tumor cells. So this is completely can be done in the lab using genetic elements that introduce using retroviral infection. So something that genetic manipulation that has been done in the lab since 1990. So it's a very old protocol applied to many human tumor models, but now we are borrowing it to study the telomere biology disease patient tumorigenesis process. So we introduce all these genetic elements that turn DC patient cells in the culture, in the plate, without touching the patient, and allowing them to become a tumor version of itself. And so far, we are successful in creating some of these models, a total of 6 of them. And I'm showing you one of them right here. So this is wild type discarin. So this is a control cells at early population doubling with telomerase put in. And then this is the same version of the cells now transformed into a tumor cells. And you can see that the morphology as well as the density of the cells are completely different. This is much more round and globular as opposed to the normal long elongated shape, which is what this normal cell type should look like. And Correspondingly, we have this Sharon patient cell line that we obtained from Sharon's collection, L54V, this Sharon mutation, along the same PDL, meaning the growth in culture age. You can see this much more sparse and much less vibrant than that of control cell type. But in any case, they're still growing, and we can actually take this cell line and also switch it into a tumorigenic cell line, meaning that they change the morphology completely. And by all these in vitro measures, are transformed tumor cells. So now we have these cell cultures. We just create a whole bunch of them, 6 of them in the lab. And now we are about to start analyzing them in using different cell biology as well as genetic assay. The number one question that we want to answer is, how does these DC tumor cells maintain their telomere versus the control cells? And comparatively, did they overexpress by increasing the copy number of either TER, telomerase, or TER, telomerase RNA, or they switch to a different mechanism of maintaining their telomere? So hopefully we will have some more exciting data to share. And with this creation of the tumor cells, we are actually very excited. So Sharon and I just sent in application for further funding to help us do the type of experiment that Brett proposed, which is to sequence the tumor genome and actually look at the expression profiling. So this in vitro or synthetic way of creating tumor could very well be complement to what Brett find with the clinical samples. And we are very looking forward to look at your data.
Wonderful. Does anyone have questions for Sinead or Brad or Judy on their research? It's a good chance while we've got them here, so. Well, thank you all very much. It was very informative, and I'm really excited to see all the work that you're doing. And again, a reminder to all of us on the call that it's the work that we're doing, the fundraising that's funding these projects and getting us closer and closer to understanding these disorders and finding pharmacology and cures and treatments that will help every one of us on the call. We're on the call because we're all touched by telomere biology disorders. We have that commonality. So it's important to all of us. And I just thank you all for presenting your research and reminding us again why this is so important and what we're here for. So again, as Katie said, you're welcome to stay on the call. This is the exciting part. This is where all the fun happens. So I'm sure you're going to want to stay. Or you're welcome to drop off. And if you drop, again, thank you very much for being here. I know your time is important, and so thanks for spending some time with us. Before you go, if you go though, I do want to tell everyone where we are with our fundraising. So we have approximately 65, Katie?
I'm just going to stop recording. So Judy, if you could make me host again, should be in my top right with the 3 dots.
So it's Team Telomere, you or?
Either one. You can, I think you can choose either Colleen or
okay.
I. And actually, I might be able to stop it.